The overall goal of this procedure is to dissect and stain Drosophila pupal ovaries.
This method can help answer key questions in developmental biology, such as those pertaining to stem cell differentiation and tissue development in the model organism Drosophila. The main advantage of this technique is that customizable tools are used to accommodate the small size and translucent nature of the pupal ovaries, which are otherwise difficult to isolate.
We had the idea to use this method when we conducted time course experiments to track stem cell division in the ovary during pupal Drosophila development.
To begin the protocol, combine 10 male and 15 female adult Drosophila flies in a vial of normal rich fly food supplemented with yeast. Allow the eggs to develop into larvae at room temperature for 3 to 4 days. After 3 to 4 days, use a moist fine brush with soft bristles to make a rolling movement with the brush along the wall of the vial to transfer wandering third instar larvae from the vial to a glass well filled to the brim with 1,X-phosphate buffered saline, or PBS. Wash the food debris off the larvae by transferring them to another well filled with 1X PBS. Separate the male and female larvae with forceps. Identify the male larvae by a pair of large, round, and translucent testes embedded in the fat body on the lateral side of the body, 2/3 down from its anterior end. Then collect at least 10 females from the well and place the female larvae into a separate well filled with 1X PBS. Use forceps to transfer the female larvae gently into a new vial of fresh fly food supplemented with yeast. Place the vial with the female larvae in a dark location to facilitate pupariation. Throughout the day, monitor the larvae for pupariation.